Hi, my name is Caitlin Chang, and this is a review of the effects of fluoxetine, a very commonly prescribed drug, on non-target organisms and the ecosystems in which they live. Pollution. We all know what it is, and these are probably some of the first things that come to mind. Garbage littering the beach, smog polluting our air. But there is one class of pollutants that no one really thinks about. And that's these guys, the pharmaceuticals. They're found in surprisingly high concentrations in our waterways, making it there through our water treatment plants. Much of it originates from improperly disposed of hospital waste, which isn't so much a problem here in the US as in other countries. People flushing their unused medications down the toilet instead of returning them to their pharmacy. And unfortunately, in our urine, either as metabolites or the original parent compound. Antidepressants are some of the most commonly prescribed medications, especially now in the time of COVID. Amongst those, according to Mayo Clinic, selective serotonin reuptake inhibitors, or SSRIs for short, are the most common. These guys aim to increase intercellular levels of serotonin, a regulatory hormone that controls mood, attention, and appetite, amongst other things, by, as the name implies, blocking its reuptake at the synapse. Amongst the SSRIs, this guy, fluoxetine, brand name Prozac, is the most prescribed and is also the most present in literature. So I started my research with a very broad question. I wanted to know what the effects of fluoxetine were on ecosystems. In order to answer this, I first broke it down to the level of the individual. I predicted that this compound might have some anti-anxiety and appetite-suppressing effects on non-target organisms due to the read-across hypothesis. This idea essentially states that pharmaceutical compounds will have the same or very similar effects on non-target organisms as long as the molecular target is conserved across species. In this case, we're talking about the serotonergic system, which is conserved across not just mammals, but all vertebrates. By that token, in theory, most, if not all, vertebrates will therefore respond to this guy, serotonin, in the same way that humans do. So once again, I predicted, based on the read-across hypothesis, that fluoxetine will have anti-anxiety effects and suppress the appetite of non-target organisms because that's what the drug does to us humans. Now, these two things we don't have to worry about, but they can decrease fitness pretty significantly in organisms that have to survive out in the wild. Since most studies focus on the effects of fluoxetine on specific species, I decided to break my research down like this. I came up with a hypothetical aquatic ecosystem, since once again, from the first place pharmaceutical wastes end up is in the watershed. In my little hypothetical ecosystem, the producers are photosynthetic microbes, such as algae. Primary consumers are little tiny planktonic creatures, such as water fleas. And the secondary consumers are teleost fishes. Let's start at the bottom with the producers. So fluoxetine seems to have an overall negative effect on these guys. SSRIs as a whole have been found to have antimicrobial effects, actually killing some groups of organisms, while others, such as algae, only saw decreased growth, smaller cell sizes, and an increase in the instance of cell deformity. For instance, in a study on the effects of fluoxetine on algae, Brooks et al. Showed that growth significantly decreases as fluoxetine concentrations increase. All of these things can be detrimental to individuals, populations, and ecosystems, as photosynthetic activity or primary production is significantly decreased. Now, a very small amount of research has been done on the effects of fluoxetine on these guys in comparison to vertebrates. So, mechanisms are still very poorly understood. Now, moving on to our hypothetical primary consumers. Remember that these are the little tiny planktonic creatures such as the water fleas and midges. Now this group overall saw reduced growth when exposed to fluoxetine. This can obviously be bad for you as an individual, reducing fitness by making you more vulnerable to predators. Interestingly, fluoxetine had various effects on fecundity, or reproduction. Some species saw increases and others saw decreases in the number of offspring produced. In the same study by Brooks et al., C. dubia, a type of water flea, saw a significant decrease in the number of offspring reared at high fluoxetine concentrations. At the same time, H. azteca saw an increase, although it wasn't significant. Now, decreases in number of offspring can obviously be detrimental to species who have hundreds of them at a time, so that maybe two can survive. But an increase in number of offspring isn't necessarily a good thing either, as investment per offspring would decrease with increasing numbers. But once again, relatively little is known on the effects of fluoxetine on invertebrates. Moving on to our secondary consumers. Remember that these are our teleos fish. It's been pretty well established that fluoxetine has anxiolytic effects on these guys. And based on the read-across hypothesis, this is expected as it's the main reason it's prescribed to humans. This has been shown in a number of behaviors. First and foremost, fish that were exposed to fluoxetine had a much slower reaction time in acoustic response tests, which simulate danger. They also spent significantly more time motionless as opposed to darting around the tank in a typical fishy fashion. More time in the upper third of the tank as opposed to hiding at the bottom. And more time in the light as opposed to hiding in the dark. All of these behaviors can obviously be very bad for survival, as it makes you more susceptible to predators. Fluoxetine also seems to have an anorectic effect on these fish. In almost every study, fish exposed to fluoxetine consumed considerably less food, and the food that they did eat, they took a much longer time to eat. This is indicative of a decrease in appetite and was unsurprisingly accompanied by weight loss. Interestingly, when the livers of these fish were analyzed, it was discovered that fluoxetine accumulates here. Hepatocyte area was significantly decreased, and less glycogen and lipids were present in the tissue. This can be seen in the tissue samples to the left from DeFarias et al., actually from this year. This one is from a fish who was exposed to high concentrations of fluoxetine. As you can see, in comparison to the control at the bottom, the cells are much closer together and not as shapely. Now, decrease in appetite and weight loss are obviously detrimental to survival, as it leads to this decrease in glycogen and lipids in the liver, which are very important for times when food might not necessarily be available. You can see now that fluoxetine has a very wide variety of effects on non-target organisms. The common denominator? It decreases fitness in all of them. Now, as you might recall, my original question was on the effects of fluoxetine on ecosystems, not individuals. Considering everything discussed here today, it can be concluded that fluoxetine has the potential to drastically alter population dynamics, whether it be by speciation, extinction, or some other means. This can lead to a number of different things, ranging from slightly different communities to total ecosystem collapse. We just don't know yet. And I know this sounds all ominous and scary, but this is just one tiny piece of a giant puzzle. First and foremost, almost none of the studies mentioned here looked at fluoxetine at environmentally relevant concentrations. They were all much higher, probably to make its effects on these organisms more apparent. Fluoxetine also isn't the only pollutant floating around out there. In real life, an organism would be exposed to thousands of compounds all at the same time, whether other pharmaceuticals or from some other source. In reality, we just don't know yet. Both of these things warrant future studies.